Hello guys, welcome to my channel. As you know, we are studying about the physiology, but we are today we are not studying about one of the topic that we decided, but instead we'll study about viruses just for one this topic, that is the coronavirus, right? As we know, it's a pandemic time, and many people have questions regarding the coronavirus, that how it came, and especially nowadays as the mutations are coming out of the coronavirus. So many people have questions that why coronavirus is mutating. Right. So before we'll go into the mutation, so first we'll see what are viruses. So the question is what are viruses. So I will try to explain as possible as that a layman can understand. Right. So viruses are not alive. First thing, in order to consider alive, any organism must have a genome and a metabolism. So that means it must also be able to reproduce and to compensate for changes in the external environment to maintain homeostasis, right? And the population of living organisms evolve over time, but not for the viruses. So viruses share several characteristics with the living things, but they are unable to reproduce independently. They also have no metabolism and do not generate their own energy. That's another difference. And viruses do not undergo cell division like living things. They completely disassembles after gaining entry into the cell and assembles new virons from scratch. So these are some of the characteristics of virus. So this is what virus is. Right. So this the first is that we know like what are viruses. Like viruses are not alive, right? And the difference between the virus and a cell or an organism, just a single cell organism, right? So that we learned today that in order to be considered an alive organism or cell, it must have a genome and a metabolism. It can able to reproduce, right? It can able to transfer the energy. So these are some of the difference between a cell of an organism and a virus. So now that we goes on the size. So the size. So the size viruses are really small in size. The smallest of the virus is 20 nanometer. That is the smallest virus. An average human cell. The average human cell diameter is 10 to 30 micrometer in diameter. Right, so that means this average human cell is 10 to 30 micrometer. Right, so that means the cells are 100 to 1000 times larger than the virus that are infecting the cells, right? The smallest of the virus is 20 nanometers, and like there are different kinds of, there are 100 nanometers virus, there are 1000 nanometers virus, right? Viruses are also obligate intracellular parasite. Viruses are obligate intracellular parasites. So that means that they are completely dependent upon the internal environment of the cell to create new infection virus particles that are called virons, right? So obligate intracellular parasite means they are dependent on the internal environment of the cell to create new infection, right? And the third point is the genetic material of virus can be composed of DNA or RNA, right? So there is genetic material in the virus, like, but the cell, normally the cell has DNA, and it has a double-stranded DNA. For the non-medical people, it has a double-stranded DNA, as you know, that has the genetic material. So this is about the cell. But for the virus, it can have DNA or RNA. It can have DNA or RNA, and it can be double-stranded. That is the short form of this DS, double-stranded DNA, or it can be. Sorry, actually it is. Oops. It can be double-stranded DNA or single-stranded DNA, or it will have double-stranded RNA or single-stranded RNA. So these are different kinds of virus categories. It has definitely genetic material for the virus, but it has no metabolism of itself. But cell has double-stranded DNA, but the virus can have a DNA or RNA as well. All right. So these are the some basic features of a virus. So now the coronavirus. So you can see the picture of coronavirus. This is how coronavirus looks, right? So it has some booking outside of the coronavirus. So what are those things, right? And we know that the coronavirus or any other kind of virus actually does not live long time on the external surfaces, like as we know on the wood or any cardboard or any clothes, right? So how long it does live, and why after some time it considered like it wouldn't infect you, right? So the reason behind all of this thing is in the structure of virus. So we'll see the structure of virus. Structure of virus. All right. So in order to be protected, virus must enter the cell. So virus cannot live outside of the cell. Why? Because if virus is even in the extracellular environment, the virus will be exposed to enzymes that could break down or degrade the nucleic acid. Right? So inside the virus there is a nucleic acid. We'll study about that as well. So physical stresses such as the flow of air or liquid could also shear the nucleic acid strands into pieces. And in addition, the viral genomes are susceptible to damage by the ultraviolet radiation, radioactive radioactivity, right? And much in the same way that our DNA is also destroyed if it is exposed to extra UV rays or radioactivity. So it's the same for virus. So that's why virus cannot survive outside of the cell. So if it's outside on your clothes, like coronavirus infection is on your clothes or cardboard, it cannot survive for a long time. For this is the reason, because it breaks out the nucleic acid strands into pieces and it cannot infect you rather. So if the nucleic acid genome of the virus is damaged, then it will be unable to produce progeny virons that will infect you. All right, so we'll draw the basic structure of virus. So for example, so this is for example the nucleic acid of the virus. So nucleic acid term for non-medical is that it's the nucleic acid. We'll write it down. Nucleic acid. This terminology is used for DNA or RNA, as virus can have either of this. So we'll just use the nucleic acid. So this is the center part of the virus, that it has a nucleic acid. So in order to protect this fragile nucleic acid from this hard environment, the virus, what it does, the virus surrounds the its nucleic acid with a protein shell. So it surrounds this with a protein shell. So how it does, it's something like this. I'll try to draw. It surrounds, so the nucleic acid is surrounded by a protein shell that is called capsid. So this is a protein shell that is known as called capsid. So this is called a capsid, right? So this is the capsid come from Latin capsa, meaning box. So this is like a box. So the capsid is composed of one or more of the proteins, different types of a protein. So this is composed of one or more different types of protein that repeat over and over again like a bricks that makes the wall, right? So this repeating structure for, forms a fit. This repeating structure forms a strong but slightly flexible capsid, right? Combined with its small size, the capsid is physically very difficult to break open and sufficiently protect the nucleic acids inside it, right? And most viruses also have an envelope surrounding this capsid. So, for example, something we'll draw here. I don't have enough space. Something like this. So this interior is our capsid, right? Our capsid, and this is envelope that is surrounding the capsid, right? The envelope is of a lipid membrane, right? Most often plasma membrane, but can also be like cell endoplasmic reticulum or Golgi. This viruses has some of, not all of them have an envelope. Some viruses have envelope. The virus that has without envelope is actually known as a naked virus or non-enveloped virus as well. So beside this, nucleic acid, capsid, and an envelope, virus more often have proteins that are called matrix protein. Virus often have this protein that are called matrix protein, and let me see if I can use. Virus often have this matrix protein. So I will try to draw with this, so maybe you can see. All right. So this are sorry. So this are matrix protein. Matrix protein. So virus. So the viruses have often this kind of protein that are called matrix protein, and the function is that it connects the envelope to the capsid. Right? That's the function of this matrix protein. Right? And as I mentioned, if it does not have an envelope, it is just called the naked virus or non-enveloped virus, right? And each virus also possesses a virus attachment protein. That each virus has that virus attachment protein, and that will show you how it actually something like this. Maybe I can draw here. So this virus attachment protein is actually protruding outside. It is actually protruding outside. So something like this, it's protruding outside this virus attachment protein. So even if it does not have this envelope, they will have this. So this are, let me write it down. Virus attachment protein. So these are virus attachment protein. So what this virus attachment protein does is the viral protein that facilitates the docking of the virus to the plasma membrane of the host cell, the first step in gaining entry into the cell. So that's the function of this virus, and this is the structure of virus. The normal structure. There are different kinds of structure. There are helical capsid structure. There are icosahedral structure. There are complex structures as well for the viruses, but this is just one of the basic structure. And if you notice, the coronavirus also have this kind of things, right? So these are the virus attachment proteins. It has the envelope, right? Right. So this is was the structure of virus. And virus classification is that virus, as I mentioned, virus is divided into DNA, RNA. So it's like divided into class 1 is like double stranded DNA, class 2 is like single stranded DNA viruses, class 3 is double stranded RNA viruses, right? These are some of the kinds of virus that are divided, and you can see on your screen the list of the virus that is infecting vertebrates. And you can see the coronavirus, I marked it, that is a single stranded RNA positive strand virus, that is coronaviridae, right? And the viruses are also divided as per the order of maybe of the discovery or of the shape or of their properties. Like for example, coronavirus is named after the its properties, like or its shape rather, as the corona meaning crown, right? So the crown like appearance of these virons when viewed in an electron microscope. All right. Okay, so now I will explain a little bit about DNA and RNA. Not in very detail, but just a basic introduction. I will try my best to just explain for the non-medical people as well. So as you know, DNA is replicated in the nucleus, right, in the cell. Like there is a nucleus, it is replicated. Like when there is a cell division, when cell makes new cell, DNA is replicated, and the same exact copy of DNA is replicated, and that is going into the new cell. So how it does? So like DNA is replicated in the nucleus. Like for example, this is cell, and in the center it has a nucleus, and in the nucleus, like as this is a nucleus, there is a DNA for the living organisms, right? So to create a copy, DNA is transcribed into a messenger RNA in the nucleus. In the nucleus, it is transcribed into messenger RNA, and the messenger RNA is translated by ribosomes in the cytosol to create a protein. Just to be simple, DNA contains hereditary information. Right down here, DNA contains hereditary information info, right? And RNA, what is RNA? So that is a temporary copy. So this is just a temporary copy of DNA gene, right? And ribosome, that creates a protein out of amino acid based upon the sequence of nucleotides within the RNA. Sorry. So I will give you one analogy. So for example, you have a desktop computer at home in your bedroom with thousands of files on the hard drive, right? One of those files is a document that explains how to complete your final class project. So the machine and the supplies you need to complete that final project are located at the school. However, instead of taking your whole desktop computer with you, you copy the single file from the desktop onto a USB drive and leave the computer at your home, right? So, right? So you take that file into the USB. So once you arrive, you take the USB to the school, and once you arrive at the school, you read the instruction and use the machine and the supplies to complete the final project. So in this analogy, your hard drive, your hard drive is your DNA, right? So your hard drive that is at your house and in the computer, that is a DNA that contains thousands of files, right? Like that's the DNA contains thousands of genes, and the temporary copy that went outside, that you took in the USB, temporary copy, that is our RNA, right? That left the house, so means that left the nucleus. So this is nucleus. For example, this is, just imagine this is the nucleus. So the RNA left the house, so means RNA left the nucleus with the temporary copy, right? And that temporary copy provides the instruction that was the temporary copy that provide the instruction that were used to create your final class project. Your final class project is a protein. So your final null project is the protein with the machine. So you went to the school, use this USB and use the machinery of the school. So the machine at the school is ribosome. So this is one of the organelle of the cell, right? And it supplies means amino acids. So the proteins are built from amino acids. Another supplies from the school, amino acids, right? That are found at the school, and the school is the cytoplasm that is outside of the nucleus. So this is just a basic analogy. So that means when we put this together, so the replication of DNA, replication of DNA is first transcribed into mRNA, that is called a messenger RNA. So that is our transcription, and this transcription is translated into the protein out of amino acids, right? So this is how, when the cell divides, these are all the functions that happens in the cell, right? Why I explain this to you? So like in this entire process, so when the DNA replication begins, so there is one enzyme known as DNA polymerase. DNA polymerase. So this enzymes actually lays down the complementary nucleotide, so that is the DNA nucleotide sequence, onto the new strand of the DNA, right? So this enzyme helps in doing that. So the DNA polymerase actually have a high fidelity, meaning that they do not often place an incorrect base in a growing strand of replicating DNA. So for example, if there is a whole entire sequence of DNA, so the DNA polymerase makes sure that there is no error in the entire sequence of the DNA, right? And even this has also proofreading ability as well. In the same way, you may type an incorrect letter on a keyboard and hit the backspace key to replace it with the correct letter, right? So the DNA polymerase can also do these things. It can reverse and replace an incorrectly placed nucleotide. DNA polymerase and there are repair enzymes can also cut out a section around the incorrect nucleotide as well. So that means that DNA polymerase makes one mistake for every 1 million nucleotide copied on an average. So this is like very perfect mechanism for making the replication of DNA, right? So that means DNA polymerase has high fidelity rate, and same DNA polymerase there is RNA polymerase, right? So this is one of the enzyme of RNA that uses the DNA template to create a strand of RNA. So this is another kind of RNA enzyme, that is RNA polymerase. And RNA polymerase do not have a high fidelity as DNA. So that means It also places an incorrect base on an average once per 100,000 nucleotide transcribed. So DNA error is 1 million, around 1 million. So the error ratio. And for RNA polymerase, it's around 100,000. So you can imagine, so the perfection of the DNA, and this is for the RNA. So RNA can make mistakes. Alright, so why I'm explaining to this? So we are getting to the mutation. So as we know, so DNA is very perfect. RNA polymerase is not perfect. So the eukaryotic cells do not contain RNA dependent RNA polymerases for the creation of the messenger RNA. This is responsible for this. So what happens? So when the virus enters into the cell, right? So the several types of virus that are RNA virus, and if you remember, we see in the category, coronavirus is RNA virus, right? So that RNA viruses encode their own RNA polymerase. So this virus, any kind of RNA virus, will make their own RNA polymerases. So virus will make their own polymerases, right? So, and especially several type of RNA virus will make their own RNA polymerases. So in the eukaryote, it is 100,000, right? So the RNA polymerases with the virus, it has an error ratio of 1 in 100, comparison to this to 1 in 100,000 nucleotides. So the RNA polymerase that the virus makes, so that makes so much of errors, and this is the reason it has a high mutation rate, because of the low fidelity of the several RNA viruses that encode their own RNA polymerase. Right? So this is why whenever virus replicate itself, so it does not have that proofreading capacity like this. And when they make their own RNA polymerase to make their own copies, so the error ratio is 1 in 100. So when they make 100 viruses, there are of course one virus that has a different copy or a mutation in there. So the mutation can be harmless. Most probably it is harmless, but sometimes it's in the favor of virus. Right, so viruses have evolved several mechanisms to ensure the successful transcription and translation of their gene products. So necessary to create more infection virons. So some viruses take advantage of the host splicing machinery to produce several mRNA transcript from one precursor mRNA. So like there are different kinds of mechanism. So the virus will use, so when there is a replication going on, so virus will interfere in one of the processes to make sure that their mRNA is processing perfect information when it is transcription and there is a translation of the gene products, right? So and there are evolved mechanism, so like it ensure their mRNA is processed and recognized by the host ribosome. Viruses has also evolved different ways, like for example some virus can interfere with the host RNA polymerase too. So like some virus can interfere with the, if this is a cell host mRNA, it can RNA polymerase, sorry, it can interfere with the RNA polymerase too. So viruses can interfere with the processing of precursor RNA. Many viruses interfere with the export of the mRNA transcript from the nucleus. So there are so many mechanisms going on for this replication of DNA to RNA and to the protein. So the virus try to interfere in any of the any of the protein or any of the mechanism and try to interfere and try to make, especially the certain viral proteins can cause degradation of host RNA, host mRNA. And what are that? For instance, this SARS, that is our severe acute respiratory syndrome that is caused by coronavirus. Caused by coronavirus. So that is our SARS-CoV. So what this does, the coronavirus, which has a protein named, and it has a protein name NSP1. So it has protein name NSP1 that induces the breakdown of the host mRNAs. So the protein of this coronavirus NSP1, it attacks the host cell mRNA and it breaks down the host RNA. And interestingly, the SARS, the mRNA of this coronavirus is protected from the degradation. So this is different kind of mechanism, and this is the coronavirus mechanism, that it degrades the host mRNA, the messenger RNA. All right. So after this, so we'll now know how the virus is replicated, the process of virus replication. So virus must undergo the process of replication to create new infectious virons that are able to infect other cells of the body or subsequent host. So what happens when the virus gain entry into our body? So there are 7 stages of virus replication. There are total 7 stages. So the first one is attachment. So this is the same as it sounds or as it means, attachment. So for example, if this is our cell, right, and this is, okay, I will make a different, like a star. This is a virus, right? So the first step is attachment. So the virus make initial contact with the cell at the plasma membrane. So this outside layer is called the plasma membrane. So what happens, this comes here and make an initial contact with the cell at the plasma membrane, and the binding of virin is a specific process that involves virus attachment protein binding to the cell surface receptors. Like the cell has different cell surface receptors. For example, like this is what it has as a cell surface receptor. So what happens, like this comes and this attaches here, the virus comes and attaches to this cell surface. So this is the first step of the virus. So virus gain entry into our body. The first step is the attachment, right? And the binding of virus attachment protein is to a cell surface receptors involve electrostatic forces, right? And after the attachment, what it does, the second step is penetration. Penetration. Right? So the second step is penetration. After making the attachment to the surface, now the virus should go inside, penetrate. So the penetration method by which the virus crosses the plasma membrane of the cell. So virus take advantage of normal cellular processes. Like there are normal cellular processes called endocytosis. So there are different processes that virus goes attachment. Like for the endocytosis means, like for example, so for example, this is some of the component that the cell needs inside the cell. So what the cell does, like it start bulging, and this goes inside and like it makes a vesicle, and so after this goes this, and like after this it will be, the star will be inside. So this is a kind of process that is known as endocytosis, and viruses use this process to gain entry into the cell, that is penetration, right? It takes an advantage of a normal cellular processes to gain entry into the cytoplasm. And this is the endocytosis is the most common method, right? But viruses use different method as well, like some of the virus use fusion or phagocytosis method, but like this is just a most common method that viruses use to gain entry into the cell. So after gaining entry into the cell, the third step is uncoating. Uncoating. So the uncoating refers to the breakdown of the viral capsid. So this is the viral capsid that we studied, right? Releasing the genome in the cell. So what it goes inside the cell, and then after entering into the cell, the next step is to uncoat itself. It's just breakdown and releasing the this viral genome into the cell. Like for our cell, our cell actually divides into two to make a new one, but for the viral, it breaks down and it comes to from the basic structure, like assembled de novo by packaging a copied genome into a newly created capsid. So you will thinking how virus goes outside. So like there are many viruses achieve uncoating uh, by escaping from the endosome that they use to enter the cell. So there is a process so that they use endosome to escape from this and then they break out. Other viruses do not completely uncoat and use the remaining capsid. So they use this remaining capsid as a home base for a replication process, and some of the viruses do not break completely. Right? So these are some of the process of uncoating. After uncoating, now the step is replication. Right? So the next step is replication. So the replication depends on the type of a virus. As we studied, like there are different types of the virus, right? Double-stranded RNA, single-stranded DNA, double-stranded DNA. Right? So I will show you just uh, the replication of the single-stranded RNA. That is our coronavirus that has. So our coronavirus is a class 4, it is written like this, positive single-stranded RNA. So this is our coronavirus comes in this class. So the RNA viruses are unique in their genetic information and it encodes using RNA. It does not have its DNA, but it encodes using this RNA. So the viruses with this single-stranded RNA genome that can act directly as mRNA are known as a positive sense RNA viruses. So this is known as a positive sense single-stranded RNA viruses. That's why it has a plus sign and it can act as directly as an mRNA. It can act directly as this, right? And similarly, like this single-stranded RNA with genomes that are not able to immediately translate by ribosomes are known as negative sense RNA. Like so, there are positive and there are negative as well. So there is a different class of virus that has a negative single-stranded RNA. But this positive single-stranded RNA that can act, it can act directly as mRNA. So it can directly act as an mRNA. So it already had a copy of its genome. So what it does, it just goes to the ribosome, to the machine, and it start making their own copies, right? So this positive strand RNA are more abundant than any other class of the RNA. So this class of RNA is the most abundant class viruses of the RNA than any other class, and infect a wide range of host species. And this include several different human viral families, including coronavirus. So the families of viruses that include this class is coronavirus, right? It has coronavirus, it has flavivirus, it has picornavirus. So there are different kinds of viruses in this families. and that cause significant disease in humans. Right, and their abundance indicate like this is very abundant, and that indicates that this RNA has been very successfully evolutionary, right? So, and the common characteristics of this RNA, single-stranded RNA, is that their infectious genome encodes a polyprotein. So its mRNA contains a polyprotein, meaning that the genome is translated by ribosome. So this goes to the machine and it translates new protein, right, into a long chain of amino acid that is then cleaved into several small proteins. So what happens? This goes to the machine, right? It makes a long chain of proteins, and then that long chain of protein is cut into very small blood proteins. So this provides an economical method of deriving several proteins from the translation of only one piece of mRNA, right? So this is one of the characteristics of this single-stranded positive strand RNA. All right, so this does the replication. This is our class for the coronavirus, and it has an mRNA. It's already had information that need to be copied and it need to be replicated. So when it is uncoated, this goes to the ribosome in the cell, and it started making their copies, right? And after that, the next step is the fifth step. After replication, it's time for assembly. So the assembly refers to the packaging of the copied viral genome with a newly manufactured viral protein. So after it has replicated, right? So it makes their own genome. Now the process is to assemble it, make like now it has just this part. Now the process is to assemble in the entire virus, right? So this can occur in the nucleus within an organelle like the endoplasmic reticulum. So there it has a number, the cell has number of different organelles. So like it goes into the endoplasmic reticulums or Golgi apparatus or in the cytosol, depending upon the virus it assembles. And after assembly, the next step is maturation. So the maturation refers to the final changes that must occur within the virus to create an infectious viron rather than an inert particle. So this process is the final step. So it makes sure like the virus that has been made, it's now matured enough to infect another cell. It's just not remain as like just a inert particle, right? So this can involve modification of cell surface. Like what happens in this? So like after this happens, like it has a cell modification, surface receptors, the cleavage of viral polyprotein or changes to the viral capsid, right? And the maturation is often tightly linked with an assembly or release. And the last process is that is the release. After that, it's time for the release. So release refers to the exit of the virin from the cell, right? And this most often occurs through the budding of enveloped viruses or via cell lysis. So what happens? So after replication, it goes to some of the corner of the cell, and like it start accumulating. After accumulating, it matures, and after maturing, it goes out of the cell, and after going out of the cell, it infect another cells, right? And the process continue, and it goes out of the host and then infect another host, right? So this is the process of virus replication in our body. All right, so this was the process of replication of virus, and the question is how virus mutates. And the coronavirus has like two mutation, or rather three mutation, from Brazil variant or Britain variant or like South African variant, and it's actually known or it has rather a name instead of calling from a countries. So what happens when all this process happens? So between all of this process, like as we know, like DNA makes sure like there is no error, but the RNA cannot make sure like the error ratio is 100,000, but for the virus RNA polymerase it has 1 in 100. So when they make different copies of viruses, like from 100 copies there are at least one that has a different kind of mutation in there, right? So mutation occurs in virus, but most probably, though it does not have virus, it's not in the favor of virus, but sometimes it can be in the favor of virus. So for example, the mutation that occurs in Britain, so maybe I'll write it down here. So that is known as, the variant is known as B117 lineage, right? And so this variant has a mutation. So this variant has a mutation. Okay, we'll write it down and complete. This variant has a mutation, where it has a mutation in the receptor binding domain. In the receptor binding domain, also known as RBD, right, of the spike protein. Of the spike protein at the position 501. The position 501, where, so in that place where the amino acid, so what happens at this position, the amino acid asparagine, so it is actually asparagine, that has been, and the initials like this, has been replaced with tyrosine. Right? So this is the mutation that happened in the coronavirus. This mutation that happens, it happens in this protein. So this is the spike protein or virus attachment protein. So when it replicates, so in this spike protein there is a receptor binding domain. So this protein has a receptor binding domain. So what it happens when it goes to the cell, it binds with the receptor binding domain. So this receptor binding domain protein has been changed at this position 501. So there is amino acid actually known as asparagine that was replaced with tyrosine. So this is the variation that happens in B117 lineage. That's our Britain variant. There is South African variant, so that is actually B1351 lineage. So this variant has multiple mutation in this spike protein. This variant also has a multiple mutation in this spike protein. Variant has been identified in Nelson Mandela Bay in South Africa. Right, so this was the different variants. All right, so that was the virus application and how virus mutates, and you know the portal of entry of the viruses, how it enters and how it exists from our body. Like when it's a respiratory tract, or like epithelial, that's the skin. And so there are different portals of entry for the viruses. Okay, and the immune response, so the immune system, our immune system is a dynamic collection of cells and proteins that safeguard the host from pathogens, right, including viruses. But over evolutionary time, however, viruses have also evolved a mechanism to interfere with the host immune response. So nearly every facet of the immune system is thwarted by some viruses, like RNA viruses with error-prone RNA polymerase, right? We know that RNA polymerase have errors, right? They can make errors when they replicate. So like our immune system has a memory that can remember, if once they kill this RNA virus, so they will remember, okay, this kind of virus, whenever it comes, we have to kill it, right? This is our immune system. But as this RNA polymerase can make mistakes while replicating, so it can make new copies, right? So they mutate quickly and can escape immunological memory in this fashion, right? So that is why sometimes it's really hard to kill this virus. Right guys, so this was the topic of coronavirus. I'll try to explain as easy as possible. Right guys, I hope you enjoyed it, and just stay safe, take care, wear mask. Right, thank you guys for watching, and don't forget